Okay, continuing on, talking about metabolism. So in chapter 1, we talked a little bit about metabolism. We're going to talk a little bit more now. So as we learned, metabolism of all of chemical operations going on in our body. Now, metabolism requires various nutrients or fuel to function, and metabolism also produces waste products. Metabolism can be thought of as all of the life-sustaining reactions in the body. Now, it's subdivided into two opposite processes, anabolism and catabolism. Anabolism is the building phase of metabolism. It's the process by which simpler compounds are built up and used to manufacture materials for growth, repair, and reproduction. Many anabolic reactions are dehydration synthesis reactions in which water is removed and biological modifiers are hitched together. Catabolism is the breaking down stage, the process by which complex substances are broken down into simpler substances. Many catabolic reactions are hydrolysis reactions in which water is added to break apart the large molecules. So again, the amino acid shows here with dehydration synthesis, it creates a larger molecule. We take the water out, we take an H and an OH from these, and it wants them to then join together. If we add the water back in, it breaks this apart and gives us the two separate compounds. Now, enzymes speed up the rate of chemical reactions, making them fast enough for your cells to use the materials. In order for your cells to be able to do anything, chemicals must be broken down and others must be made. You need building materials to build the small cell particles called organelles and to make energy. Any of these processes require chemical reactions to occur in the cell. The problem is that when these reactions are very, very slow on their own. So we need something that's going to speed it up. That process, those solutions to getting those to go faster are special proteins that we know as enzymes. They have enzymes are protein molecules that have special binding sites on them that will help speed up a reaction. Now, one of the things about enzymes is they're used in the reaction, but they're not used up in the reaction. So they just facilitate. They'll bond to the enzyme molecules bond to the biological molecules and work through the reaction, and then when the reaction's done, the enzyme goes back and picks up more. Those are called substrates. Because enzymes are a binding system, they are specific. Only certain molecules can be carried by certain enzymes. Substrates can compete for binding sites. If all binding sites are full, enzymes are said to be saturated. Molecules must wait for an empty enzyme before going through the reaction. So in a way, if we think of the helper, think of an enzyme like a sponge, okay? So you can soak up all of these substrates until your sponge is full. But until you squeeze that sponge out and all the product is gone, you can't pick up more in that sponge. But through the whole thing, the sponge never changes. All in all, the sponge is still a sponge. So think of the enzymes kind of like a sponge. Enzymes can be blocked or inhibited, preventing the substrates from binding, and enzymes generally are unchanged by their participation in the reaction. So this illustration is out of your book, and it shows that we have an active site available. This is our enzyme. So the substrate, our sucrose, is there, and it jumps on board, and it goes for a ride, and it comes down, and hydrolysis occurs, so the enzymes assisting this action from happening. Let's think of enzymes kind of like the streetcar, okay? You can get from one end of Canal Street by the river all the way up to Mid-City if you walk, but it'll take you a long time to get there. So you hop on the streetcar. Well, and the streetcar gets filled. Until the people get off the streetcar, they can't pick up more. So There's another analogy to help you out with the enzymes. But the streetcar stays the same.